like to start by thanking the organizers for inviting me here. It's been a long way, but it's really great to actually be with you today. So I'm going to try to discuss a bit about another side of the story in terms of the metabolic complications. And a lot of the work that we're going to be presenting actually stem from investigating long-term effects in childhood cancer survivors. So I agree that there's still quite limited literature at the moment actually describing long-term consequences of irradiation, but there are a few papers here, and noticeably is one of the papers that was actually published with one of my collaborators, who actually set up a cohort of long-term survivors in Australia. And in 2006, they actually identified that having been treated as a kid for cancer would actually predispose you to developing different kind of metabolic complications, and in particular here, hyperinsulinemia, impaired glucose tolerance, and type 2 diabetes. When I actually pushed a bit further to try to identify the mechanisms, what I identified was the fact that receiving total body irradiation was actually the main risk factor, even higher than receiving bone marrow transplant. So clearly here it seems that there's something happening that's linking receiving irradiation to developing long-term complications. We can also see that there's been some additional reports, and in particular for the A-bomb survivors, but we've actually heard about those papers previously, and there's also radiation exposure in mouse models that have also shown that you can actually develop skeletal muscle function, and metabolic activity can actually be impaired in response to irradiation. So that actually gave us the idea to try to investigate what could be the leading mechanisms here. And to address that question, what we did was to develop a model of irradiation, a mouse model of irradiation. And so basically here we had C57 black mice that were actually exposed to 6 rays, and that was the work at the time of a PhD student from the University of Copenhagen, Wiebe Nylander, and that was done in collaboration with the team of Professor Romain Barès, who was also in Copenhagen. And so what we did, we exposed those mice to 6 gray. That was just one hit. We let them recover for 5 weeks, and then after that, the groups were actually split between a high-fat diet and a normal diet group. And we actually monitored them on the long run. What we found was that very early on, there was a delay in weight gain, which is very consistent with what we've just heard before. So basically what we found is that irradiated mice here, so those who were on a chow diet, but also those who were on a high-fat diet, were actually characterized by a lower body weight. And basically, when we had a look at what was driving that decrease, there was a delay. 1 week after irradiation, mice that had been exposed to irradiation didn't gain any weight. And that delay, they never recovered. So that means that at the end point, they had decreased body weight. And that was also associated with a decrease in body fat, as well as a decrease in lean mass, which is mostly muscle. 20 weeks post-irradiation, or 25 weeks post-irradiation, we then investigated whether or not there was an impact on the capacity to actually regulate glucose. So what we did, we just injected the mice with a bolus of glucose and actually investigated how quickly they could actually clear that excess glucose from their circulation, and that allowed us to actually measure an index of insulin resistance, the OMA-IR. And what we found was the fact that if you combine irradiation with a high-fat diet, you actually get an increase in insulin resistance. So it seems that on the long run, having been exposed to irradiation and combining this with a high-fat diet might predispose you to developing irradiation, in particular compared to those who are only exposed to a high-fat diet. You probably all know that, but there are major organs that are involved in the regulation of blood glucose in our body. Under postprandial conditions, so like us, we just had a coffee and probably a couple of sweets, about 85% of that excess glucose that ends up in our circulation will actually end up in our skeletal muscles. So when you start developing insulin resistance, when you start developing hyperglycemia, it means that your muscles are not taking up that glucose anymore. And as we've heard in a previous talk, that's actually tightly regulated by insulin signaling, which is actually cascaded through AKTPI3 signaling. In addition to skeletal muscle, adipose tissue can also actually act as a sink for that excess glucose, although they cannot contribute as much as what skeletal muscle do. As I mentioned to you earlier, skeletal muscle is quite important in regulation of blood glucose, and in particular through insulin signaling. So we investigated whether or not that defect in terms of glucose uptake was actually linked to a functional impairment. And so when you take skeletal muscle and when you extract the cells, you stimulate them with insulin, you can actually see an increased phosphorylation of AKT. Now, what we found is that upon irradiation, that response was actually blunted, and the same happened in the high-fat diet group. When we extracted stem cells or satellite cells from skeletal muscles, and when we cultured them, we also found that they had a lesser capacity to proliferate. And as I explained previously, they also retained that phenotype that I described in the whole body, in the whole animal. They still retain that phenotype, showing that they had a defect in terms of taking up glucose. So you take satellite cells, you extract them from the mouse, you culture them, and they retain that metabolic phenotype that we observed in the mouse. So that's actually suggesting a long-term memory effect. Some of the key mechanisms that are involved in actually perpetuating that memory effect are epigenetic mechanisms. So that actually led us to interrogate whether or not there could be changes in the epigenetic signature within those tissues. And so again, we extracted the satellite cells, and we ran a whole genome balsified sequencing, and we then had a look at different genes and the different level of methylation. What we found was that irradiation actually induced changes in methylation level in over 4,500 genes. When we ran a gene ontology analysis, what we found was an over-enrichment in pathways that actually involve in metabolic pathways, but also in cell cycle regulation. To try to further identify what were the leading mechanisms, what we did, we developed an in vitro model where we then monitored one of the key enzymes that's involved in the regulation of methylation level, which is a dimethyltransferase. And so when we measured the activity of DNMT1, we didn't find a difference, but what we found was the fact that there was an increased cytoplasmic translocation of DNMT1, which could actually explain why we actually found that remodeling of the epigenome within those mice. I explained previously that not only muscle is important in glucose regulation, but adipocytes, the adipose tissue is quite important too. And so Nadia, in collaboration with Luca, who was a Danish student who came to do a master's in Australia, they actually developed a model where we could actually interrogate the function of pre-adipocytes. So again, you digest your adipose tissue, extract the pre-adipocytes, culture them, and try to interrogate their function. And the key function in adipocytes is the accumulation of fat. So when you differentiate them, they tend to accumulate a lot of lipids. And this is something that you can actually monitor using flow cytometry. And what we found was the fact that irradiation actually induced a decreased capacity to differentiate in pre-adipocytes, in particular when they were not exposed to high-fat diet. We also looked at insulin signaling through the monitoring of phosphorylation of AKT upon insulin stimulation, still by flow cytometry, and we found that there was, again, an effect of irradiation. So clearly here, those cells are no longer functional, and even when you actually extract them from the host, they still retain that memory of the host metabolic profile. So again, we actually ran a whole genome analysis to detect methylation changes. We found in excess of 3,000 genes that were differentially methylated between non-irradiated versus irradiated. And again, we found an enrichment in metabolic processes on top of the list of our gene ontology analysis. Some of the key mechanisms that can be associated with impaired activation of DNMTs is inflammation or oxidative stress. So we then try to profile infiltration of leukocytes within adipose tissue. And what we found was that high-fat diet led to an increased proportion of CD45 positive, so panleukocytes if you want. But more interestingly, what we found was a particular enrichment in M1 macrophages. So it seems that there is more of a pro-inflammatory profile taking place within the adipose tissue that could actually explain that remodeling of the epigenome. A couple of years ago, we published a review where we tried to actually link changes that are happening within the immune system that could actually explain metabolic defects, and in particular involving epigenetic changes. And we know that the immune system is actually quite tightly regulated through many different epigenetic mechanisms. Methylation is one of them. Histone modifications or microRNAs are quite important in that context as well. And we know that if you change the epigenetic profile of immune cells, that will have functional implications. And we know that in the context of obesity, for instance, you get an overrepresentation of Th1 cells, but also M1 macrophages, and all those processes are actually tightly regulated through epigenetic mechanisms. So to try to see whether or not our results could actually be transferred into human, we recruited a cohort of long-term cancer survivors, childhood cancer survivors, and that was the work of my PhD student, Sarah Daniel, and my current PhD student, Brianna Clifford, in collaboration with Viber. And so we recruited patients who had received only chemotherapy versus total body irradiation plus chemotherapy. We then sorted lymphocytes, and in particular here, CD4 T cells. These are the only results that I'll be showing here. And we performed representative reduced bisulfite sequencing, as I showed in the previous slides. And what we found, again, was the fact that there was in excess of 450 genes that were differentially methylated in CD4 T cells from irradiated patients, in irradiated survivors. And there was a particular enrichment in immune processes, which is not that surprising when you're dealing with CD4 T cells, for instance. But more interestingly, there was an enrichment in oxidative stress and inflammation as well. We then investigated whether or not those changes were actually linked to functional impairments. And what we found is that if you take those PBMCs from survivors, you put them in culture, stimulate them with a mitogen, what we found is an overactivation of Th1 cells in the irradiated group versus not so much in the non-irradiated group. So what we found is that upon stimulation, we had overrepresentation of CD4 T cells producing interferon gamma in the total body irradiated group versus the other group. When we looked at whether or not this had some consequences in terms of systemic level of cytokines, we found an increased level of plasma interferon gamma, TNF-alpha, but also IL-2. Interestingly, we were able to actually recapitulate the findings of our collaborators more than 10 years ago, in a sense that within our population, we found that patients who had received TBI had increased level of LDL cholesterol. They also had increased level of total cholesterol. But in addition to that, they tended to actually show hyperglycemia, and they had increased insulin resistance. So here you can see that excessive immune activation within irradiated patient is actually linked to a change in their metabolic profile that might predispose them to type 2 diabetes. Nowadays, if you don't touch on the microbiome, you're not cool. So we had to actually interrogate part of that. And in particular, what actually drives us to interrogate the contribution of the gut microbiota is the fact that the T cell epigenome is also under the control of changes within the gut microbiota, and in particular within the gut. So we know that there is a clear communication between the immune system, and in fact, most of our cells actually get educated within the gut, do so under the influence of the gut microbiota. And we know that dramatic changes in the bacteria within our gut will actually drive a change within in the profile of the cells that are actually present. And the profile between Th17 and Tregs is actually quite tightly regulated. And we know that in the context of obesity at least, changes in the gut microbiota or dysbiosis can actually lead to impaired structure of the gut and increase the gut permeability, which can actually lead to a state of metabolic endotoxemia, which will then lead to immune activation. And that's a definitive contributor to the inflammatory component that we can actually find in metabolic diseases. So that led us to the hypothesis that potentially irradiation is also actually driving that immune activation through changes happening within the gut microbiota. I got lucky because I prepared a summarized slide before I even got told that I had to have one. So this is pretty much trying to summarize in a nutshell, but also raising some hypotheses about what's happening when we get exposed to irradiation. So potentially one of the key mechanisms that's happening upon irradiation is that change that's happening in the gut microbiota. That change in the profile of the bacteria that we have in our gut, referred as to dysbiosis, might actually be leading to that impaired function of the gut, which will then be associated with increased leaking of LPS and other type of bacteria within the circulation, which will then drive an activation of the immune system. We found in our study that there was increased mobilization of pro-inflammatory leukocytes within the adipose tissue. We also found that it was actually associated with an epigenetic remodeling of the stem cells within those tissues, and that was also associated with functional alterations, namely here, insulin resistance. Similarly, within skeletal muscle, we didn't investigate whether it was actually linked to inflammation, but what we found nonetheless was the fact that there was, again, remodeling of the epigenome within those cells that was linked to functional alterations, insulin resistance, and that was also associated with impaired mitochondrial function. To finish up, I would like to thank all my collaborators within UNSW in Sydney, the Sydney Children's Hospital, the Prince of Wales Hospital, the University of Sydney, as well as my collaborators from Denmark and my collaborators in Hiroshima in Japan. Thank you for your attention.
Are there any burning questions from the audience? Yep. Sorry. It's okay. We have time to.
So, and did you see any change, linking it with the previous talk, did you see any change in IGF-1, or is this not relevant to this system?
We didn't measure it.
Okay.
So we have complementary information on the role of the metabolic system.